Just a brief introduction about MPS. MPS NOVA is a twinning project led by ITQB and NOVA Medical School, both part of University of Lisbon. MPS NOVA aims to establish an European hub of excellence in microphysiological systems, MPS, fueled by the pluripotent stem cells technology to strengthen UNL research and innovation capacity in the area of chronic disease and host microbiome interaction. MPS NOVA is designed to promote knowledge transfer among partners, to leverage NOVA University full potential to translate basic research in chronic and infection disease, host microbiome relationship into clinical applications. MPS NOVA-driven partnership of international leading institutions will enhance the existing multidisciplinary knowledge and expertise of the NOVA MPS Hub, advance our best practices in our research and training, and expand collaborative networks. And if you want to know more about the MPS project, you can follow us on our social media and also on our websites. And now, without further ado, I will introduce our first external speaker. So today we'll have two external speakers, starting by Cláudia Almeida. She is Chief Scientific Officer of CEO Lab AccelBio, where she leads scientific strategy and bridge academic biomedical research with the industry-ready drug discovery. With over 25 years of experience spanning R&D and academia and GMP regulated environment, she has held senior roles River Laboratories, Ireland, and build a strong track record in methods development, lead team leadership, and grant acquisition. Internationally, experiments across Portugal, Ireland, and Brazil. She holds a PhD and has been a PI and collaborator in multiple EU-funded research programs. About Ricardo Viais, he is the Chief Operations Officer at AccelBio, where he manages operations to ensure its sustainability growth. With a chemistry and neuroscience, Ricardo holds a PhD in biomedicine from University of Barcelona. After his PhD, Ricardo shifted his career to RI management, working with national and international projects with a common goal, contributing to advance biomedical research and promote the robustness of R&D institution and worldwide. And today they will talk about the AccelBio, accelerate drug discovery to turn ideas into investable and impactful assets. So the stage is yours, and you can start when you want.
So I will start, then I will present myself, then Cláudia will present herself, and then we'll talk a bit about AccelBio, a bit what we are as a collaborative laboratory, what we do, and then we have some slides where we try to go and present how we can bridge with your twinning project, because we have several models of organoids, which I think can lead to some synergies and some mutual learning here. So, but to start with my career, so Juliana already said a bit, but I started by doing a degree and a master in biochemistry at University of Porto. So there, between Faculty of Sciences and ITQB, Instituto de Ciências Biomédicas Abel Salazar. So both the master and the degrees between the two institutions. And then I did an Erasmus and a master thesis already in the field of neuroscience at Universidade Autónoma of Barcelona. I went back for 1 year to Portugal, where I was in a research grant. But then I looked for a PhD, and I went again to Barcelona, but to a different institute, where I did a PhD in biomedicine in the field of uncovering cellular pathways of embryonic neurogenesis. And after that, there was a big question mark: what will I do after? I was looking for some postdocs also, but at the same time, I was doing a postgraduation in project management. And it was by that time that I applied to a twinning project in Portugal. So I returned to Portugal to be a project manager of a twinning project like yours, which is called EpiNet. And the idea was to find partners across Europe to strengthen the capabilities within IMM, Instituto de Medicina Molecular, in the field of epilepsy. So that was what I was doing for 3-4 years. And then getting more experience with project management. Then in 2024, mid-2024, I enrolled. So after the end of the other project, I started at AccelBio as a project manager. We had by then 3 projects ongoing. Right now we have around 11, 12 projects. And since the beginning of 2026, I'm Chief Operations Officer, means I get a bit more responsibility on leading what is the general operations of AccelBio, how we organize, how we present ourselves, and but still and managing a bit the team of project managers. That's what I do. So I will pass to Sylvia. She will present herself.
So hi everyone, good afternoon. It's a pleasure to be here today. Thank you for your invitation. So very briefly, I started my career by doing BSc, a previous licenciatura at Faculdade de Ciências, Universidade de Lisboa. I then moved on to the previous IGC, which is now GIM, and I obtained my PhD in 2007 through ITQB, Universidade NOVA, on the field of virology, immunology, focusing on uncovering viral mechanisms of immune evasion. After this, I went through 2 postdoctoral fellow cycles, let's say one at Faculdade de Medicina Veterinária, Universidade de Lisboa, and another one at IMM, which is now GIM, following on the path of unraveling viral mechanisms of immune evasion, and at GIM focused mostly on uncovering immune regulation pathways. In parallel, I was also a lecturer at multiple institutions, lecturing mostly immunology. And then in 2017, looking for a very different challenge, I went to Brazil, where I stayed for 2 years as an associate visiting professor. And then I moved to Ireland because I wanted to come back to Europe because I was very far away from home, where I joined initially as a virologist and then progressed into the last position I had, which was in vitro development manager, operating under GMP environment. More recently, in September 2023, I joined AccelBio as Chief Scientific Officer, and I've had the pleasure of leading and dealing with a very talented team of scientists across multiple fields, and we're going to discuss a bit that today. Ricardo, yeah, you can proceed.
Okay, so talking a bit about AccelBio and what we are, I wanted to take a look here at the bottom at this. Well, it's not the most beautiful scheme, but it shows a bit what comes from, if you can see my mouse, what comes between the discovery of an idea that generally can occur in academia and the entering of a drug, of a product into the market after phase 3. So we have the preclinical phases, and we know that many very good ideas that are found and discoveries that are found in academia and startups, many of them don't even reach the preclinical the clinical trials, even though they have a very good potential. And this is known as the valley of death, where many drugs fail to go through. And they fail to go through for several reasons, some of them money-wise, some of them regulatory-wise. And what AccelBio tries to do, and why it was born as a collaborative laboratory, is to support entities from academia or startups to go through this valley of death and empower their potential drugs or their potential targets, going through target validation, hit ID and hit ID and validation, lead optimization, and ADMET studies, and also going a bit into preclinical. So we don't go into clinical trials. If we have a very good idea, very good assets, we, in principle, don't take them to clinical trials. Either we license it out or we create a spin-off. These are the several options. But our mission is to guide and support the transformation of scientific projects into successful drug discovery programs ready to be invested and that could deliver novel therapeutics. So we started our operations around 3.5 years ago. So we are pretty young, and we count with, so here again, we work collaboratively as I said, to accelerate preclinical drug discovery, serving as a bridge that brings biomedical science closer to the market. And we don't work alone, but we work with our associates. We were born with a set of associates that help us to deliver our mission, and these include three academic entities where we have our laboratories based. Biocant, which is a science park located in Cantanhede. We have some startups and pharma companies, and we also have a VC with us. It doesn't mean that they fund us. It means that they can guide us, and if there's something that is very good, they are the first ones to know about it. Okay, and we count a lot on these associates. So we start there already collaboratively. So how do we work? Always in a collaborative approach, and we can work in several ways, several collaborative models. So in collaboration, either through strategic partnership, through by joining competitive funding. If you want to contact us, and we receive several contacts for projects where we enroll into consortia and we apply for competitive funding. We can also have risk sharing strategy, and we had some open calls where we for 2 years we selected projects to support. We give some money, and more important than that, we give our translational support, as I will show you later. But also we develop platforms, and we can also provide tailored services where an entity, either from a research center or a private entity, comes to us and they want just a set of experiments done where they own all the IP, everything gets on their side. We are just providing a service, and that is also possible. To which kind of entities we work with? Universities and research institutions, and universities and academic institutions, research institutes, startup and early stage biotechs, and pharmaceutical companies. And we have worked with most of these cases in the projects. So this is our now external collaborations that we have ongoing, or that we always extendable. And this is our team. Sorry, because you are sometimes in front of us. This is our team. We are now 20 people. So 22 people. Some of them are PhD students that enroll our team. We are a multidisciplinary team where we have people with experience in translational research coming from startups, from academia, from bigger pharma. We also have a big team on the organoids field, on computational medicinal chemistry, bioinformatics, proteomics, in HTS, and here actually there's a very strong know-how on host interactions, host pathogen interactions, and we also have the innovation manager. So this is our team, and we operate under 4 strategic pillars. There used to be 3. Now I added a bit the 4th. You understand why. One is the acceleration of drug discovery projects. I will explain this a bit, how we work with teams to accelerate drug discovery projects. We have a set of projects that we have. We are quite agnostic about the field, but we have some tools to accelerate these projects, and I will tell you later. Then we also develop some platforms, so technological platforms that on one hand can support these projects, and on the other hand can help us to provide services on its own, okay, or to enroll into consortia, for example. Then we also have training programs where we have webinars in different areas, and we also support through non-academic PhDs. We already support around 15 PhD students at this point. And this is also a pillar on us, is to provide training to train the next generation of entrepreneurs and translational researchers. And last but not least, we also have we aim to build a strong biotech innovation ecosystem. Focused in Portugal, we have an ecosystem of biotech that it's growing, it's becoming robust, and it's based on very good basic science that we have. And we either we attract events to Portugal, or we participate into international consortium working with other clusters to try to strengthen our biotech ecosystem. So in the first pillar, acceleration of drug discovery projects, so we evaluate targets and concepts according to industry standards, and with that we count a lot with the experience of Sylvia and other people in the team. We establish a multidisciplinary team. We design proof of concept studies around new targets and candidate products, and we use our expertise and network to tackle different aspects of drug product discovery and translation. So important for that, for every project we support, we match them with one of our translational researchers so that they can help the project, build a roadmap and a roadmap to the market and support these projects. We also have an innovation manager that can also support these projects. And how? By helping teams in experimental design, and in some cases in performing some experiments, in helping teams in designing a target drug profile, which is a very important tool for a drug and to test and to analyze how a drug is doing on its way to market. We also do market analysis, development planning, we help with regulatory strategy and with business planning. So on the other hand, and now Sylvia will enter, I also told you we have a research and innovation agenda in key emerging technology platforms, and the platforms that we have is the advanced in vitro systems, the organoids that we're going to present today, the computer-aided drug discovery. We're just going to talk about it very briefly today, but we also have, based on our associate capabilities, HTS capabilities, and multi-omics capabilities. Okay. So, Sylvia, the floor is yours now.
Yeah, just to give you a highlight in relation to our technological platforms, more specifically our organoids. So we have two platforms, heart and kidney organoids, which are proprietary. And so in terms of what characterizes them, or kind of an overview of the model, they are derived from human iPSC. So they are complex in their structure and they are full 3D. They are physiologically very relevant because they recreate multiple cell types that we normally see in the human organs while integrating. So, but it's not just a recreation of or a representation of the cell type. So we maintain molecular significance, structural significance, and the ability to have functional features that are clinically relevant. So they are predictive because they have the ability to provide human relevant data that can help accelerate preclinical decision making. Their engineering is, let's say, simple because it doesn't rely on any complex addition of any particular scaffold or extracellular matrices, which can oftentimes be the cause of great variability, and they are scalable for screening and compatible with 96 well plate formats. So Ricardo, if you can, yeah, in terms of how we can use and apply our models, so they can be used for disease modeling, target and compound screening, toxicology and efficacy studies, teratogenicity studies, and also developmental studies, because we can recreate different stages of development of both the heart and the kidney, so from embryonic to adult. In terms of where we stand of our proprietary model, so we have 2 PCT patent applications submitted, and we are now working through FTO studies for the kidney, and we have this finalized for the heart. So in terms of where we stand in comparison to other competitive platforms, so naturally, as I said, our organoids are quite complex. They fully recreate 3D structure for both the heart and the kidney, with the maintenance of structural architecture and physiology. If we compare in relation to these 4 or 5 parameters, so our organoids stand out because they are HIPSC derived. We have two different organoid models that can be used for this type of studies. So they recreate human physiology, both from a point of view of cellular complexity, but also from a point of view of organ architecture and physiology, or readouts, physiological readouts. And we can also mimic or study different stages of development from embryonic to adult-like. And as we progress, so in terms of the overall complexity of, or the type of cells or structures that are represented in the heart and the kidney, for the case of the heart, we can have or develop different types of organoids where we have represented ventricular myocardium, proepicardium, coronary-like vascularized epicardium myocardium organoids, and also sympathetic innervated myocardium organoids. And so I would like to highlight the fact that one of our differentiating features is the fact that we have indeed vascularization and innervation for the case of the heart organoids, for the case of the kidney, which we can develop as a 2.5D or full 3D. On top of the complexity, I'm so sorry, we have also vascularization, and we have represented the main architectural units, which are podocytes, proximal tubules, distal tubules, and also glomeruli. We also have strong expertise with brain organoids, as well as the ability to generate other types of organoid model depending to the requirements of any given project. But again, these are not proprietary compared to the heart and the kidney. Ricardo, if you can. Yeah, in terms of the heart organoid, jumping a bit into the cell composition and the structure, as I said, we can recreate or demonstrate physiological relevance and replicate the varied cell type differentiation, structure, and function. And we have all these different cell types represented. So epicardial cells and epicardium and subepicardium space. We have fibroblasts, endothelial cells, accumulation of adipocytes. We have a myocardial area where we have compact myocardium, smooth muscle cells, and also pericytes, as well as sympathetic neurons. Yes, and so this gives you a more visual image of what I have just said. So where we can actually see, if we go to the top left region of the slides, we have represented the myocardial zone, the epicardial zone. And if we zoom in into each of these areas, you can see for the case of the epicardium, the mesotelium, the subepicardial space, and then we enter the myocardial area. And if we zoom into the myocardial zone, we see a distinction between a very compact myocardial area, and as well as the representation of the different types of cells that we can actually see in these organoids. So CD31 highlights vascularization. Lipid droplets we can see on the middle bright region, we can see as green, and it gives a good highlight of the complexity of these structures. Additionally, it is also possible to recreate the sarcomere structure, which is the fundamental repeating contractile unit or functional unit of striated muscle, as is the case of the cardiac muscle, and which allows for the measurement of contractility in these organoids. And if we progress, yes, in terms of the predictive value or how these organoids can be used for the case of toxicity and safety evaluation, more specifically cardiotoxicity. So these organoids are now validated for two big classes of drugs. So drugs that have been withdrawn from the market due to cardiotoxicity, because it was not possible to predict from animal studies, and also anti-cancer drugs. I would also like to highlight the superior predictive value of our organoids in comparison to the use of just human cardiocytes, be it from HIPSC-derived cardiomyocytes, or even from adult ventricular primary cardiomyocytes. And in the next slide, this is what we show. So for the case of cisapride, so a drug that is normally used for the treatment of gastroesophageal reflux disorder, we can see that when we apply it or when we expose heart organoids to increasing concentrations of these drugs, it is possible to induce irregular contraction. So we see an irregular frequency of the contraction, and we can distinguish between an early after depolarization and a delayed after depolarization, because we can have the induction of a irregular frequency, and then the way the organoid recovers from this frequency of contraction can have early effects or delayed effects, which in the case we see both. And if we compare this to the HIPSCs derived cardiomyocytes, we see that the use of this more simpler cell line does not allow...
Eu também já podes tirar, que eu já tirei essa questão.
Yeah, so and this is something that is not recreated by the use of these cardiomyocytes alone. And if we continue, so in terms of another type of core service that we also provide, or it can be a service or a partnership, is to use these organoids in disease modeling studies. And this is being done for cardiomyopathies, particularly for two different types. One where we are looking into arrhythmogenic cardiomyopathies, and we are participating in EU project where we are assessing the role of and the contribution of the epicardium to the mechanisms underlying this disease. And in parallel, we are collaborating with a Portuguese pharma company to assess some small molecules in terms of their safety and efficacy in a model. So it's cell lines from patients of hypertrophic cardiomyopathy to enable lead identification and prioritization. And we are talking about things that we have now ongoing. In terms of the kidney organoids, similarly to what we have described for the heart, cell composition and structure is very representative of what we find in the human organ. So this, for the case of this organoid, our results have been validated by microscopy and RT-PCR analysis, and we have now ongoing validation by bulk and single cell RNA sequencing. We can recreate the major physiological units in the kidney, naming collecting duct, distal tubule, proximal tubule, Henle's oops, podocytes. We have also the renal stroma represented and the basement membrane. If we dive into the architecture of the organ, so this gives us a representation of all the physiological units that I've just described, namely proximal and distal tubules, collecting ducts, renal stromal with podocytes in the periphery, the Henle's loops in the center of proximal tubules, and then the renal stroma with the basement membrane around it. We can also detect, as we can see to the right, and as the organoid matures, more specifically around day 60, we see maturing glomeruli that develop across the glomerular basement membrane in the middle of the podocytes. In terms of the functional features, that we can assess with the kidney organoids, we can do nephrotoxicity studies, we can do water transport and cyclic AMP, like the case of phospholipid-induced cyst formation, and we can also study tubular transport, namely through dextrin and albumin uptake. And if we continue in terms of the predictive value of the kidney organoids, so we have validated our platform for toxicity and safety evaluation, for nephrotoxicity, for anti-cancer, analgesic, antibiotic, antiviral, immunosuppressant, and drugs that were withdrawn from the market due to nephrotoxicity. And we have compared the performance of the kidney organoids against immortalized human proximal tubule epithelial cell line. And what we see is that for the case of KIM1 and LDH, which is a measure of tissue damage marker, we see that the organoids, in comparison to the HK2 cell line, are markedly superior, and we also see that the kidney organoids allow for detection of clinically relevant markers that are related with renal injury. And even looking at the panel of immunofluorescence where we have a live dead assay represented, we can clearly see that in relation to the control, the various drugs that were tested induce cell death, particularly prominent in the periphery of the organoid, as well as a distortion of the classic, let's say, architecture or structure of the organoid. And if we continue, so in terms of opening windows of opportunity for additional studies, so a lot of effort has been put into maintaining organoids in culture for more extended periods, and we have gone now until day 90 with maintenance of the architecture and also of the physiology, and this opens up a window of opportunities for studies, for example, in diabetic kidney disease and also chronic kidney disease. Progressing. Okay, I don't know if you want to take over now, Ricardo, or... Yeah.
It's the same. I can also just finish. So just present the second, the other platform that we have. It's our last slide, our computer-aided drug design platform, which we have. We can do experiments on target validation. So this is in silico, target validation, hit identification, lead optimization, and early ADMET studies. You will have access to the slides. But important, we can do drug discovery, protein discovery, and engineering bioinformatics, and we have our own computational capabilities based in Cantanhede that allow us to have our own computational resources to work with the partners that we have. And I think that is on us for today. We extended a bit, but if you have any questions, we'll be happy to answer.
Thank you very much, Ricardo and Sylvia. It was very nice to meet your company. So does anyone have a question? Yes, Sandra?
Hi, right. It was great to know you better. I have several questions, but well, first of all, regarding toxicity of drugs that are first metabolized in the liver, are you addressing that as well in your platform?
So for the moment, that is not a capability that we have directly. No. So we have done some preliminary work on liver organoids, but not to the point where we have them integrated as of yet in the platforms that we offer. Yes. Mm-hmm. And that would be one of the main ones to really look into because of its value and in terms of excretion, processing of drugs. Absolutely. Yeah.
It would be definitely something interesting in the context of using sheep, and I think that.
Yeah. Very nice. And sorry, maybe I was distracted. Do you have a library of compounds, your own libraries?
No.
Not yet.
No.
Okay.
Not at this stage. No. We normally work with, I'm sorry, Sandra, what we normally do, even with the partnerships that we have ongoing, is that the sponsors or the clients, so it can be a collaboration or a fee for service, they would supply the drug that they want to test if it's their own drug, or we can source the drugs and get them so we can do the studies that are needed. Yeah.
And your platform is a high throughput screening platform with organoids, or medium? Will say medium throughput screening?
Yeah, I mean, I would say medium, because what we know we can do and we have tested and optimized is 96 well plates. Now, this will depend on how many drugs you want to test, how many concentrations, all of this, because we know that at this stage, with the conditions we have optimized, our organoids, both heart and kidney, do not deal well with too much handling. So if we place them here and there, they really suffer, and we cannot downscale too much the physical space that they have, because they are very huge, they are very big, and so they will suffer tremendously. And at this point, 96 well plate is the smallest they can handle.
Yeah, very nice, very interesting, and it's great to know that we have this in Portugal, in Cantanhede, near my town.
Well, in this case, actually, organoids, so the organoids themselves, they are developed at Tagus, in Oeiras. Okay. But where we have the HTS capabilities, that is being developed in the laboratory of our collaborator Miguel Mani, which operates at Biocant.
Okay. Nice to know. Great.
Thank you, Sandra.
Thank you, Sandra. I don't know if anyone has a question. So if not, thank again Silvia and Ricardo, and we will move to Cláudia Almeida. Okay. So just a briefly introduction of Cláudia. So Cláudia Almeida is a scientist, a group leader at Nova Medical School in Nova for Health, where she studies the cell biology of brain aging and Alzheimer's disease. Her research focuses on how intracellular traffic, endosomal pathways, and synaptic dysfunction contribute to neurodegeneration, and her laboratory combines cellular, molecular, and imaging approaches in experimental models to understand early disease mechanisms and identify potential points for intervention. And today, she will talk about developing human neuronal microphysiological systems to study early mechanisms of Alzheimer's disease. Thank you again, Cláudia, for accepting our invitation, and the stage is yours.
Thank you. I will share my screen now. I have to say while I share that I haven't seen you, Silvia, since the university. Hi.
Hi, a long time ago. I think I saw you at the IGC or ITQB, I think.
Okay.
Or GIM, I don't know. Portugal is a very small place. Yes.
Okay. I was recognizing your face, and then when you said about me, then I realized, okay, this is where your face comes from.
Nice to see you again.
Nice to see you. Very nice. Okay. So I'll be kind of not taking too long because we are already running a bit over time. And so I will tell you a bit about why we are interested in Alzheimer's disease. And of course, because as you know, as we are living longer, the number of cases has been increasing. And since there's no treatment or cure, there's a huge socioeconomic burden. So what is important to me and why I think that it's important to know the early mechanisms is because indeed Alzheimer's disease starts many years before the onset. As you can see here in this scheme, 0 represents the onset, and biomarkers for amyloid start occurring first, even 20 years before the disease. So this means that the pathobiology occurs even without symptoms. And what causes this these early mechanisms that in Alzheimer's disease? So most of what we know comes from modeling the familial early onset Alzheimer's disease genetic forms, and these are mainly linked to genes that have to do with beta amyloid production, the amyloid precursor protein, and the 2 presenilins that cleave A beta out of APP. And by making experimental mouse models and now also organoids has been able to recapitulate that the introduction of these genes do drive beta amyloid accumulation and tau, and eventually cause neuronal loss and at least some memory loss in the mouse models. But actually, these represent only a small percentage of cases, and the majority of the cases of Alzheimer's disease, most people we know develop Alzheimer's disease after they're 65 years old, and we do not know what is the cause. We know that aging, polygenetic risk factors, and environment are important factors. So this is a complex disease, and probably there's not one Alzheimer's disease, but several subtypes. And what we are interested in understanding is exactly how these genetic risk factors contribute to the disease. At least that's what I'm talking to you about. And when I started my lab, as I'll show at the end a little slide about my career path, we analyzed the genetic risk factors, and we found that 1/3 of them are actually endocytic regulators. So we decided to dissect the function of two of these, BIN1 and CD2EP. And initially, we took a knockdown approach in mouse primary neurons, and we tried to identify the mechanism. So these are just some highlights from our findings. So in this slide here, focusing on CD2EP, so we discovered that the loss of function increases beta amyloid production through increased processing of the amyloid protein, and this actually contributes to synaptic loss. In terms of mechanism, we saw that the defects in APP trafficking can be detected even at the level of one endosome, where in control cells, APP is in the lumen of the endosomes, but without CD2EP gets retained on the endosomal membrane, and this is now thought to be a main spot for the processing and production of beta amyloid. And more recently, we found that CD2EP also regulates the actin cytoskeleton in dendritic spines, and without CD2EP, we lose dendritic spines. So this indicates for a postsynaptic function for CD2EP, linking the endosomal trafficking to defects in excitatory synapses. So for BIN1, actually, it seems that it's most important in regulating the trafficking of base, not of APP, but it does impact APP processing, and it also leads to intraneuronal beta amyloid. And it does so not by regulating sorting within the endosome, but by regulating recycling. So without BIN1, base gets trapped in endosomes, and in fact, these get larger, but we also have observed that within synapses, they become smaller. Especially in inhibitory synapses, we found that BIN1 is enriched in this synaptic type, and this leads to selective loss of inhibitory cells with consequences to the network activity related with hyperexcitability. So this is where we are now. And so why do we want to model late onset Alzheimer's disease in human neurons? So not only to understand the mechanisms, but we also want eventually to get to therapeutic targets and to test drugs for in-patient derived neurons. So this is where we see the future going. So right now, what we are doing is that we are using CRISPR to introduce mutation variants of these genes in healthy iPSC cells so that we have isogenic controls and mutant ones, and we are differentiating them into 2D neurons and assessing the phenotype. So I'll briefly mention how we are doing this. So we are using this NGN2 induction system, and actually to obtain excitatory forebrain-induced neurons that are mostly glutamatergic. And then we have different transcription factors for inhibitory neurons. We have already managed to differentiate inhibitory neurons and excitatory neurons. So next step was to achieve synaptic maturity. So as you can see here, by 28 days, we start seeing synaptic puncta along the dendrites, and at D45, the density increases. So I have some more readouts that we are using. So we are looking at the complexity of these neurons using shoal analysis. And what you can see here is that with time of differentiation, the complexity increases. We can also measure beta amyloid levels in different compartments of the neuron. So here we are comparing the soma and the dendrites. And interestingly, you see that the increase with the differentiation is higher in the dendrites than in the soma of these neurons, which is suggestive of what we know, that synapses are important for APP processing. We've started to characterize the endosomes and also the APP localization in these human endosomes, which we can see here that they are at the correct location. And these are our synapses. So by 45 days, we now managed to reach a good level of maturity of synapses with opposition of synapsin and PSD95 along the dendrites. And as you can see, the number of synapses increases with time of differentiation. For the inhibitory neurons, it's still more preliminary, but the positive things is that we managed to have and detect inhibitory synapses positive for VGAT, which are also positive for synapsin. And we can see BIN1 colocalizing with VGAT in some of these synapses. And we also saw, as with excitatory neurons, we saw that inhibitory neurons also develop more synapses with maturation time. And this is where we are now. I have some more data, but it's still confidential, so I cannot tell you about it, but it's quite exciting. So what we are working on as well now is to increase the complexity by doing co-cultures of excitatory and inhibitory neurons. I forgot to say that we add astrocytes for support. And this is our future direction. So a little bit like with the AccelBio, we want to go into more complex organoids to do drug testing, but also to recreate aging by inducing inducing senescence in our cells. Eventually, we would like to, in the future, as I said, to be able to have this platform running for patient donors so that we could, really apply precision medicine on them and test the best therapeutical option for each person's brain. So this is my take-home message. And I really want to thank MPS for the support. It's been great to be part of this community, and this has helped us to build these human neurons platforms and to bring closer to translation to human cells our discoveries from mice. So now, last but not the least, so I was asked to include here something about my path. So as Sylvia, I graduated in biochemistry in 1999, so last century already. And then I did a master in neurosciences at the Faculty of Medicine. That's when I started to get interested in the brain. And it also got me to plan my PhD in biomedical biosciences with the specialization of basic neuroscience, which I defended at the Faculty of Medicine, but I actually did all my research in New York. And that's when I started to go deep into Alzheimer's disease and got very interested in this connection between endosomes and synapses, which I'm still interested in. So of course, all of this was possible thanks to funding. And as you can see here, FCT has been very important during my path. So with the PhD fellowship in the beginning. Then for my postdoc, I actually got 2 fellowships, one from Marie Curie, one from EMBO. And this really allowed me to do postdoc in Curie in France. And here I got dedicated in studying trafficking and actin cytoskeleton. Then thanks to FCT again and the investigator FCT program, I managed to become independent and to start my lab at CEDOC at Nova Medical School. It was also very important to get again money from Marie Curie Actions with the reintegration grant. And since then, I've been working. So I kind of divide these 2 phases of my lab of building the program. And so here I was funded after the FCT investigator by FCT SEC individual, and then more recently by principal investigator SEC, which it's still my current status. And in the meantime, I've been acquiring very important funding, both European funding from JPND for neurodegenerative diseases, national awards, American funding, once again FCT, and more recently from La Caixa. So these are some of the outputs from the lab. So we've been managing to publish really interesting papers. So if you haven't had the chance to read, I really suggest you do, because I'm still really excited when I reread them again. And very important, and maybe even more important than the publications, are the people that have passed the lab. So I've now trained 6 PhD students and 10 or more master students. I'm sorry if I've forgotten someone, and 4 postdocs. In the meantime, I've also gotten some awards. One that I'm quite proud of is this Blended Learning Award, because this is for teaching. And since nowadays there's been this huge discussion about, do investigators know how to teach, and should there be only professors or professors and researchers? I have to say that I think it's very important to teach as scientists. And I think you cannot teach if you don't do science. So it's very hard for me to distinguish these 2 types of careers. So finally, my lab. And of course, I have an older picture here because I really want to thank Anton, that was the pioneer of setting in the lab the human iPSC cells and neurons. I mean, George worked actually initially on the CRISPR and the iPSC cells, and then Anna, which is currently in the lab, working on the inhibitory human neurons. So thanks a lot for listening. Hopefully it was not too fast, but there was not a lot of time left. So that's as good as it gets. But I would like to have some questions or feedback.
Thank you so much, Claudia. It was a very nice presentation. Thank you for letting us know more about your work. So does anyone have a question for Claudia?
It was all very clear, I assume.
Yeah. Sandra?
Claudia, very nice work, and it's very nice to see evolving. I know that it's not easy to, and I know that you tried already to induce some aging phenotypes in these neurons. What are the other, because you mentioned it, that as a aim, how you are thinking now to address it? I know that you tried a strategy with the nidylation. Are you exploring?
Yeah, we gave it a couple of tries. So we haven't tried a lot, right? So I mean, we haven't done as much on this as I would liked. So right now we don't have any dedicated funding for this aging experiment. So I think we are going to we have to try it more consistently. So we gave it a couple of tries with this incubation with an adylation inhibitor, and the cells died. So I think we need to optimize the concentrations and time of treatment and see. Because, I mean, the feedback that I have from Agnieszka is that she's been using it in organoids and it works. So it could be just a resistance of scaling up the concentrations and things like that.
Could be 2D versus 3D, different susceptibilities that...
I mean, in the papers, it has been used in 2D. So I think it's just maybe the culture conditions that maybe our cells were not as confluent or resistant, and maybe if they are more isolated, they are more sensitive. I mean, neurons are tricky.
Yeah,
So,
yeah. But are now you going to brain organoids?
depending on funding.
Ah, okay.
Yes, so we are waiting to know the results on the La Caixa. So we got to the interview. So if it's positive, we'll get into organoids. If it's not positive, we'll continue to write it as future aims, as we've been doing for the last 10 years.
Well, you have done a lot of things in the last 10 years.
I know, but it's been 10 years since I've been proposing to do organoids, and it's still not there yet, but we'll get there.
Yeah, sure. Very nice work. Congratulations.
Thank you.
Thank you, Sandra. Does anyone have a question? Okay. I don't think so. So thank you so much, Cláudia. It was a very nice presentation. And for accepting our invitation, Cláudia, also Silvia and Ricardo. And I hope to see you all in our next MPS seminar in July. Bye.
Thanks a lot for the invitation. Bye-bye.
Thank you very much for the
Thank you.
invitation. Bye.
Thank you.
Thank you.